Very hello students. In this way I am coming on UNACADEMY platform for CET classes, but actually I am taking NEET for everyone. Okay, because in NEET only all the state exams, medical exam has been included. So all of you can join me in the UNACADEMY classes and you can avail the facility. I take special classes student especially for all of you because all the classes are absolutely free. You people need not to pay a single rupee from your pocket, and you can utilize completely the classes, and it will be very useful for every one of you student if you follow the classes. Similarly, every day I am taking classes for the students on UNACADEMY. Generally I follow the timing, that is from 3 to 4 and 8 to 9. 8 to 9 is a fixed time students, I'll tell you. Night 8 to 9, 8 p.m. to 9 p.m. is a fixed timing I take classes for UNACADEMY students, and that will be very beneficial. So what I have started doing this, I'll be recording the sessions and putting on YouTube, but if you come for a live class, it will be really interactive and useful for all of you. So if you come for the classes, this will generally help every one of you, and this will be properly guiding you through. It will be a guide post, it will be guiding all of you and will be helping you to attain good marks in the coming exam. So let's start it, let's crack it in this way. Prepare nicely for the 2021 or class 12 student as well as class 11 student. We are going to prepare in this way, and I make sure that all students are going to get good marks. So all the best and all of you, and with UNACADEMY, let's crack it. Thank you all of you, and we'll meet in the next class students. Okay students, am I audible to every one of you? I'm sure I'm audible to every one of you. Yes, I'm audible to every one of you, I guess. Yes, okay. Okay. So in the last class, we have already seen, let me add one more slide and we'll start with that slide first. Okay. I'll just add on one more slide. That's the beginning slide, no? It has to be there. Okay, yes. So the beginning slide is there. This you see students, you can go for a plus subscription also. One of the very best plan what we are all having is you can go for a plus subscription. And it's a good subscription, you know, it is very useful for the students. Subscription is very useful for the students, you know, and it has got a lot of benefits what we can see over there. Yeah, there is a n number of benefits when we go for a plus subscription, and it is very beneficial for all the students. That is, you get a very premium content. All of you are getting a very premium content available to you. Also the structure of the, if you see, the structured live courses and dedicated live doubt clearing will be there. Any doubt you have will have live classes as such what we are having right now, and such kind of live sessions will be done for your doubt clearance also. So that is really beneficial for the students. That is really beneficial for all the students, we find. Okay, so that is one very basic criteria which you find in the live classes, and which is a lot helpful for the students also. Along with that, we'll have a lot of quiz sessions which will be arranged for all of you for the self-analysis. We'll also have batches for the completion of the preparation. That is what we can see, and test series and DPPs, the full length test and mock test and analysis, and many things you people can work out with this particular program. This is going to be quite helpful for all of you. Has everyone understood this part? So the subscription, if you see, then it is of various prices you are available. What I would suggest is to go for a 12-month plan. Why? Because it is, you see, it's quite cheaper. Because if you go for a 6-month plan, it is around 10,125 rupees, which is quite high price, I feel. And a 1-month plan is much more higher, to 2,250 rupees. But if you go for a 12-month plan, it is hardly around 12,375, and as well as you have a catch in it. What is the catch in this particular thing? That is, if you use my code, if you use this code, a 10% of discount will be given to all of you. If you use this code, a 10% of discount will be given to everyone. That is what we are going to find, student. So I would suggest all of you to go with the 12-month subscription only. That is always good for all of you, and it will definitely help you. So that is how it is. Okay, student. Now I want everyone to, let's go to the next slide. We have already discussed about this. We'll go to the slides. Many of you have already done with the classes. Okay, so we'll start with that part. Let us now go ahead. Let's now go ahead. Okay, yes. Now, without wasting time, we'll go into the slides now, all of the slides. So there are many slides which we are going to discuss today. Okay, there are n number of slides which we are discussing today, and we'll try to learn about it. So I guess the last class we have already done. The last class is already done, which I remember. We have already done with the bulbils. Okay, bulbils was the part which we had completed, and the example was agave. Also, we have done with the phylloclade, that is opuntia. Phylloclade, this is opuntia. Acha students, corona is quite much prevailing in your, in the complete India, pan India. It is prevailing like anything. Yes, good evening, Ashish. So it is prevailing like anything, pan India, students. I would suggest you, all of you, please do take care of your health, students. Do not go outside. Do go with the mask only. That is only one very mandatory things which you have to do. Please go with the mask only and do not go outside. Basically, the theme is to avoid as much as possible to go out. Do not go out. Stay at home. Stay safe and stay healthy. That's very important, students. Every student is supposed to follow that. Every student is supposed to follow that. You can interact with me on the chat box. Okay, my live YouTube sessions are happening now. So you can join me on the live YouTube session also. Today also I had one session. The live YouTube session will be from 10 to 9. I'll let you know whenever there's a live YouTube session. On 12th, 12 to 9, again we have a live YouTube session. On 12th, again I have a live YouTube session. So I would ask you all to join and take the benefits. A lot of questions I'm discussing in that live YouTube sessions. Okay, so from 12th September, there is a live YouTube session from 10 o'clock, 10 a.m. to 11 a.m. 10 a.m. to 11 a.m. I am using that live session. Every time I am going for the live sessions. Okay. So that is one very important fact, students. Shall we go ahead now? Okay. So the next is artificial method of propagation. That was very important. Root is one of the way, that is the cuttings of the root. Different types of cutting can be done through which the artificial method of propagation can be utilized, students. And that's one of the very best method through which when the cuttings of the roots are going to be used and it is going to be utilized, it will be very helpful for the every individual. That is going to be useful for all individual. Let's see how. When we go to the root cutting, we can easily see that in case of the root cutting, students, okay, the cuttings, when we find the root cutting which is going to be performed, students, will be helpful. For an example, where do we use it? And that's a question. We use it in radish. Okay, root cutting is basically used for citrus fruits. So you see, under citrus fruits comes lemon. Okay, then we have got oranges. Let me make orange with the orange color only. Oranges. Blackberry is there. Yes, we have got the blackberry where we are going to use the root cutting. Raspberry is also there. We are going to use the root cutting. Raspberry is also there where we are going to use the root cutting. These are some of the very good aspect where the cuttings of the root can be utilized, student. That can be also used. For an example, you see the root cutting in case of the radish, where it is going to be performed in the first picture. How the root cut is sliced into small pieces, that we can see, and these sliced pieces will be again put into a pot with the soil. It will be sowed and will be put into the soil. That is what we are going to see, students. Shall we go next? Stem cutting is also one of the method which we are going to follow. Where do we have the stem cutting and how we are going to use the stem cutting is, you see, okay, where do we have the stem cutting and how we are going to use the stem cutting? That is done in this manner. That is, okay, now you see the use of the stem cutting can be done, students, or just of the stem it can be done. Okay, cutting should be around 6 to 10 inches long. The cutting has to be around 6 to 10 inches long, and between the nodes, it has to be put between the nodes. That is one very important fact which we are going to see. You see one of the flower pot. In this flower pot, as you can see, so in this flower pot, the leaf internode can be seen. I'll just zoom in so that it will be a little more convenient for all of you to look into. So when we zoom in a little, we can see, yeah. So leaf internode can be seen over here, students. You see, I'll just see. The leaf internode points are there over here and here. And this is the complete leaf internode cross section what we are having. And the cutoff of the leaves with the flush of the stem. You see how nicely the leaf has been completely removed. It has been cut off flush with the stem. And then in the next step, we are going to see, and at angle of 45 to 60 degree, there is an angle cut which is going to be made. So at an angle of 45 to 60 degree, there is an angle cut which is going to be made approx 1/4 below the internode, 1/4 below the internode. And now you see the plant is kept in a rooting hormone. Now what is the rooting hormone which we are going to use? Auxin. Auxin is one of the very good rooting hormone. If the auxin is in a higher quantity, auxin is used for rooting purposes. If the auxin is in higher quantity, let me tell you, student, auxins are used for rooting purposes. So it will be cut, it will be put in a solution with a good amount of auxin. And through the auxin, as the rooting takes place, then the complete peg is going to be inserted. The complete peg is going to be inserted into the growing medium, as you can see. So thus we find that, what we are going to see, we find that one of the very best method of the artificial method of the propagation is said to be stem cutting. In plants we are learning right now, students. So in plants we are going to learn that, and double then and place the cutting into the soil. So after dibbling it, then it is placed inside the soil where the root already which has appeared will be utilized. Root which is already appeared from the stem can be utilized. Let's go to the next slide and see. Leaf cutting can also be seen. You see the leaf cutting can also be seen. Many of the plants you find that the leaf cutting can also be utilized. It can be used. A small leaf pieces, okay, leaves are used for reproduction of the new plants. That can be done. A small leaf pieces will be utilized, and the cuttings will be used for the formation of the complete leaf that can be seen in the students. Okay, have you all understood, students? That can be easily seen. One of the very easiest and one of the very frequent method which is going to be utilized. That is what we are going to find. Okay, now how to use it, how to have the benefit of it, let's see. You see the stem is cut. Okay, after the cutting of the stem, the parent plant, this was the parent plant as you see, and this is the cutting. Okay, with the help of a blade it is going to be cut, and this cut portion will be inserted inside the sand, soil or compost, so that the rooting should take place. Always remember, student, for this rooting to happen, it has to be inserted deep into the soil. It has to be inserted deep into the soil, students. Excuse me. And it should have root promoting hormone should be present in that. The root promoting hormone should be present. So this soil, if it has a root promoting hormone, let me write it over here. Okay, if we have a root promoting hormone over there, then it is going to be utilized. Okay, students? There if we have a root promoting hormone, definitely it is going to be utilized student. That can be easily utilized. So these are some of the very good cross sections which we have seen till now. You see, so it should have a root promoting hormone. Let me write that also, so that it will be useful for every one of you. So it is going to have a root promoting hormone. Write it over here. There is, it has a root promoting hormone. Root promoting hormone. Okay, that is one very important fact which we are going to look into, students. Okay. Let's see next. So artificial methods is one more artificial method comes out to be is the soil layering. We call it as a soil layering or mold layering. Soil layering or the mold layering can be seen. Okay, what is soil layering and mold layering? Let us try to understand. So the soil mounded to the cover of the base especially are pruned to the young trees, and now this is referred as a stool layering also. We also call it as stool layering. Now the question is, where are we going to use this concept? We are going to use this concept in case of apples. Okay, it can be used in case of pear. Okay, it can be used in case of gooseberry. Now one of very important point is that most of the plant which we are going to use this layering technique are going to be winter season plants. Most of them are winter season. Soil layering winter season plants mein mil jata hai. You see how properly a mound layering can be done. You see how the mound layering is being prepared. How the mound layering is going to be prepared, students. You can see over here. Mound layering is going to be prepared properly in the soil level. Okay, and the complete structure. We find that the complete plant is growing through the soil, students. It's going through the soil, students. You can see over here, right? Yes, through the soil. Now the root will start appearing, students. Now the root will start appearing. From where does it appear? From the part of the plant which is beneath the soil. So I'll just zoom it for all of you so that it will be more easily visible. Okay, so the part of the root which is inside the soil, you see from the root small rootlets are appearing now. From the root small rootlets are appearing, and this is something which is miraculously happening. Now when the stem is cut from this part, it can be easily rooted into a next complete, that is the portions. Okay, the next complete portions, it can be easily rooted, student. So these are some of the very important things which we see, that is the root arises from the stem part. I hope everyone has understood, students, right? Good. Let's go to the next. Asexual reproduction continuation we are doing, that is gutti formation. What is gutti or air layering? What is gutti or air layering condition? We'll see that. Interrupt cambium and covered wound with a moistened medium, generally used in Ficus elastica and magnolia. In Ficus elastica and magnolia, these all plants, basically we find that the gutti or air layering technique can be definitely utilized. Okay, it can be definitely utilized. You see, with the help of a blade or a scalpel, the cut is made. One layer of the complete structure is going to be removed, and after this removal, this area becomes completely open, where we are going to provide with this hay stack. Straws are going to be provided over there, and after providing such all medium, it is going to be covered with the help of the plastic. It is going to be covered with the help of the plastic, student. And after the covering is completed, then you see it is wrapped properly. Okay, it's going to be wrapped properly and will be left, from where the roots are going to arise, students. From there we find that definitely the roots are going to arise seriously. Next, you see very good example of Guti formation has been given, and let us try to see that. A very good example of Guti formation has been done, and let's try to learn how Guti formation is going to happen. Let's learn about it. You see, with the help of a plastic, it is covered, as you see in the diagram. With the help of the plastic, the Guti formation has happened. How nicely it is going to be, completely covered with the straw or the haystack. It is going to be covered properly, okay, and then it is going to be utilized for the purpose of the root growth. Then we can find that the roots are going to come out of it. It helps in the root growth, student. Root growth purpose. Root growth purpose, it is going to be used, students. One of the very best example what we are seeing right now. I hope till here it's understood to every one of you. Shall we go ahead? Okay, good. Now we'll go to the asexual reproduction, another that is called as types of layering, simple layering. Let's see. One type we are going to learn. One is called as simple layering. What is simple layering? Let's see. Low hanging branches which are covered with the soil and with or without wounding. Sometimes it is not put, any wound is there, not any kind of scalping is not done, wound is not put, and in that way we will find that the complete plant is going to be used. That is one best method of simple layering. Example comes out to be jasmine. Basically we are going to use it for the shrubs. We are basically going to use it for the shrubs. You see, either it will be little wounded or without wounding also the bend is made. You can write C over here, that is the bend is made, and through this bend we are trying to, the one which is beneath the soil, this portion will be utilized for the growth of the rootlets coming from this area. The rootlets from the bend beneath the soil. Also it can be by wounding it and then putting inside the soil. That is called as simple layering technique. This is one is called as the simple layering technique. I hope students, everyone has understood this concept. One of the very easiest concept one can figure out. One of the very easiest concept one can figure out, student. That is one very important part which we have done. Now we'll go to the next part. Let's see. Now when we go to the next part, we can see the next diagram comes out to be serpentine layering. Serpentine layering means in a complete fashion within the soil, the serpentine layering is going to be done. You see how serpentine layering is done? The layering in which at several places the stem is put inside the soil. At several places we find that the stem is put inside the soil. What is the benefit of this kind of serpentine layering? Let us try to figure out that also. The benefit of serpentine layering is that in serpentine layering, at multiple level the roots are arising. At multiple level the roots are arising. I hope you are able, all of you are able to follow. You see, at multiple level the roots are arising, and the stem has to be very flexible, student. That is one very important part. The stem has to be very flexible. If the stem is flexible, if the stem is highly flexible, then only it can be used properly. So the stem has to be very flexible, student. That is one very important criteria for such kind of stem layering, serpentine layering, which is going to be done, committed. Okay, I hope till here everyone has followed it and has understood. Serpentine layering, that is how we use. Okay, so one of the very important part, example come out to be is clematis. One of the best example comes out to be is clematis. One of the best example comes out to be is clematis. It's heavily raining over here. How is the weather in Bangalore and other parts of Karnataka and other parts? Students, can you tell me? In my place, it's a heavily raining, you know, it's a heavily raining. What about Rachna and other students? How is the weather over there? Is it Raining over there also, or it's not, it's a sunny day? What kind of weather is there? Okay, because no, you have to take care. If it's raining, do not go outside, do not get drenched into water. That is not good for you presently this time, because exams are so near. That's the reason. Let's go to the next part. Okay, so we'll go to the next slide. Now there is a condition that is called as a trench layering. As the name suggests, the layering is going to be in the form of a trench. Like a trench, the layering is going to be done. We call it as a trench layering. So it will be like a trench. So what is a trench? Underground, when we make a tunnel, that is called as a trench. Whenever there is an underground tunnel kind of structure which we make, that is called as a trench. Example, we use it in walnut. So basically we see that trench layering is generally done in case of walnuts. Walnut we do it. Okay, another is mulberry. Now mulberry is, can anyone tell me the botanical name of both of the plants? Can anyone tell me the botanical name of both of the plants? That is mulberry as well as the walnut. Mulberry is, walnut is Juglans regia. Juglans regia. Okay, and mulberry is Morus alba. Mulberry is Morus alba. Morus alba is a mulberry plant. So these are some of the plants which are going to be utilized, student. These are some of the plants which are going to be utilized, and that is how we find that the trench layering is possible for all such plants. Trench layering is possible for such a plants which is going to be taking place. Okay. Then mound layering is also there, one of the method that is called as mound layering. What is mound layering? You see, there is a soil mounded to cover the base, and especially it is pruned in the young trees. Also preferred as a stool layering. Again, where are we going to find these structures? They will be seen in case of apple, they will be seen in case of pear. In pear also we are going to find that. Pear also we can see, and also they can be seen in the gooseberry. Gooseberry. So these are some of the plant, as well as jasmine also we can find definitely. Jasmine also we are going to find it. So such all plants and all we find that the complete, that is the layering, is quite possible. So apple, pear, gooseberry, jasmine, some of the plants are there where the mound layering is quite possible, student, and we can go for it. Mound layering can be done. Have you all understood? I hope everyone has understood. We'll go to the next part. What we have, let us try to say in the same slides. Artificial method of layering, one is also called as the soil layering, also called as a tip layering. In that, tip layering is one very important method which we are going to follow. How does tip layering occurs? Let's try to figure out. In case of tip layering, the tip of the plant is going to be put inside the soil by folding it, and from the region where it has been put, we can find that the arisal of the root starts in such a plant. So the root starts arising in such a plant. You see, this is called as tip layering, where we can easily see the tip of the plant has been put inside. The tip of the plant has been put inside, and from the tip is arising. The portion which is put inside the soil, the portion which is put inside the soil, from there we find that the roots are arising, students. So root arises. There we find that the roots are going to arise, students. So you can easily figure out that is the root is arising from the part of the portion of the plant where the tip is being completely molded inside the soil, and from that part we find that the arising of the root takes place. One of the very important method which is used, which is going to be used for the artificial propagation in case of plants. So artificial propagation of plant is possible through this method only, student. These are some of the methods through which it is going to be propagating inside the soil. I hope everything is understood. Every part is understood. Now students, what I am going to do is, there are many students who are studying in class 11. There are many students who are studying in class 12 also, attending the classes. So I am going to blend the classes, 2 classes every day. I am going to take. Every day I take 2 classes. All of you know the timing. The timing is in afternoon from 3 o'clock to 4 o'clock and from 8 o'clock to 9 o'clock. These all are in PM, students. At this particular time, okay? In this particular time slots I am taking the classes. Everyone knows about it, right? I know that everyone knows about it. So what we are going to do is in the evening also I will be taking, anyways I'll try to wind up with the chapter today if possible, and then again we are going to learn class 11 portion also from the other days. Let's go to the next part. Now, asexual reproduction by grafting is also possible. There is another method where we are going to use, that is asexual reproduction, where it is going to be through the grafting. Let's try to see how grafting is going to be useful for the plants, for especially the formation of the complete, that is the root, and a new plantlet can be formed. Now for this, let me zoom in and we'll be able to see properly. Now you see there is a plant, okay? So this is a plant with a desirable trait which we have got. So this one is the plant which we have got with a desirable trait. What we wanted, that trait is present. But we have got a small root system which is an undesirable trait. Flowers are good, that is desirable trait, but the root which is small is an undesirable trait and we don't want it. We want to remove this particular. So what we do is we go for a grafting. Means choosing a plant with a very good root system. We are going to choose a plant with a very good root system, students. Here what we are going to find, students, let us try to see. Yes. In this condition, what do you find? The graft is prepared with the stalk and scion where the cut is going to be made. So there will be a proper cut which is going to be made. The lower part on which it fixes is called as stalk. And the part which is supposed to be grafted is called as scion. It is supposed to be grafted. Now, in this graft which is being performed over here with a deep rooted system, a more number of the flowers can be easily arranged. With a deep rooted system, more number of flowers can be easily arranged. So you can easily see, students, see here we have got a small flower, another plant was there which was an undesirable trait, that is having a plant with a small flowers, but the root system was very good. This was one of the desirable trait which we see, and this was the undesirable trait what we are going to look over here. So these are very easy processes through which the organism can easily have a grafting, one of the very prominent method, very good method through which the plant are going to be produced. Let's see some of the example under that. So the grafting is done. Grafting can be done in many plants, but the most famous for grafting comes out to be is something which you gift on Valentine's Day, and the answer is grafting is famous for roses. In roses we find that it is very famous. Grafting is very famous in case of roses. Okay, rose, the grafting is performed and it's very famous among the roses, students. Now let's see what we have in the next part. In the next part, students, you see whip and tongue grafting. Now what are the different types of grafting we are going to learn? That is what one we are going to follow now. That is whip and tongue grafting can be possibly can be done. What is whip grafting and what is tongue grafting? Let us try to see. Number one, let's see. So a plant with a root system, and you see how it is going to be cut in the section. It is going to be cut into the section. Again, we find that there are two scion in stock. One scion is there, one stock is there. That is how it's going to be produced. One scion is there, other is a stock. And now with the scion in stock, we find that the complete plant is going to be put into, together, and will be tied with a thread. It is going to be tied with a thread. As it is tied with a thread, the cambial activity starts. So tying with a thread initiates the cambial activity. So what happens in such a situation? We find that it is going to initiate the cambial activity. So it initiate cambial activity. It is going to initiate the cambial activity, and this cambial activity, that is xylem and phloem activity, is very important for the growth of the scion. So cambial activity is important for the growth of the scion. We are going to find students. I'll write it over here. Important. Cambial activity is important for the complete growth of the plant. For the growth of the plant, we find that the cambial activity is important, students. Now, asexual reproduction, there is another technique that is called as a bud grafting. क्या है ये bud grafting? कैसे होता है? What is a bud grafting and how does it occurs? कैसे होगा ये bud grafting? आइए इसको हम समझने की कोशिश करते हैं. Bud grafting is also one of the very important methods, students, which we are going to use. Okay, bud grafting is also supposed to be one of the very important methods which we are going to use. Let's see how this bud grafting is happening. Now you see already there is a bud. We are going to cut and remove these buds, okay? And a bud of our own choice we are going to put into. Budding we are going to do with our own choice. You see, with the help of a blade, the cut is going to be made to the C, D and E, and then a new bud is inserted. A bud with a desired quality, a bud with a desired trait, a bud with a desired phenotypic, morphological expression is going to be completely grafted over here. And after grafting, we find that the bud is grafted in such a way that there should be a cambial activity. The bud is grafted in such a way that there should be a cambial activity, and thus the bud starts growing. Let me tell you, students, that we have one more class today, that is from 8 p.m. to 9 p.m. 8 p.m. to 9 p.m. we have one more class, students, and we are going to meet again in that class also. 8 p.m. to 9 p.m. let me tell you, we have one more class which is the continuation of this class, whatever is left, so that we are going to complete, students. Okay, good. Now we go next. That is another is the artificial method, that is micropropagation. Now this artificial method, students, what we are going to see is going to be done with our own sources. Okay, they are going to be done with the help of our own sources. We are going for the artificial micropropagation, and it has to be done in a lab condition only. The important part is it has to be done in a lab condition only. If we do not follow the lab standard measures, then it cannot be done. The lab standard measures, if we have not followed, then it cannot be done. So let's see what are those and how it is going to be seen. Generally it is done in orchids. Okay, gladiolus, you see all these plants have a very bulky leaf stem. Bulky in the sense means thicker. Chrysanthemum, okay, chrysanthemum is a flowering plant. Anyways, gladiolus is also a flowering plant. And carnations. Carnations are basically used in bouquets. We gift. Carnations are very expensive plants, you know. Orchids and carnations are very expensive flowers which are basically used in bouquets. When we gift the bouquets to our friends and relatives, carnations and orchids comes out to be one of the most expensive flowers. Okay, orchids and carnation comes out to be most expensive flowers in the complete bouquet when we give to any person. How is a micropropagation done? That is done with the help of a condition that is called as, we call that condition to be as that is P, T, and C. We call it as a PTC. What do you mean by PTC? That means plant tissue culture. We call it as a plant tissue culture. Through the PTC, plant tissue culture is going to be used. A very beautiful diagram is mentioned over here. You can see there is a very beautiful diagram which has been mentioned over here. And through this diagram, where the sectioning of the carrot is being done. This is a carrot plant. What is the botanical name of carrot? That is Dacus carota. So in Dacus carota, we find that it is going to be the part will be taken in the form of an explant. Explant will be put into a nutritive media which forms a callus, and then through that callus the plantlets. If the cytokinin is more, then leaves originates. If auxin is more, root originates. Cytokinin shoot originates. So in this way we find that the plant will be growing, and as the plant grows, students, as the plant germinates, embryo is formed. Embryo will be put into a complete, that is a lab condition in a test tube. As it grows further more, then again it will be put into a lab condition only, but inside a soil and the pot. Soil and the pot, we are going to put it inside that only. I hope every student is able to figure out, every student is able to understand. Are you all able to understand? Now summary what we have studied today. So what we have studied is asexual reproduction, we have already seen, and you were all with me who have understood. Then vegetative propagation is done, natural method and artificial method. There is a two method of vegetative propagation. That is the natural method and the artificial method can be performed. Have you all understood? Okay, good. So please, all of you definitely go for the plus subscription of the CET for 2021. If you want all such classes, also you'll find me in the plus classes also, where I'm going to be useful, resourceful for every one of you for attaining good marks in your test. Shall we go more ahead? Let me load another video, another PPT, which will be dealing with. Just give me a moment, student. I'm doing. Okay. There are many methods through which the sexual reproduction in flowering plant can also occur. Okay. Sexual reproduction, you see, it requires syngamy and complete, that is conjugation. Syngamy can again be of two types, that is the endogenous, that is the endogamy, and the exogamy. Endogamy and the exogamy. Through these methods we find that it can be inserted. Isogamy, anisogamy, oogamy, hologamy can be seen. We'll learn all the terms individually. We are going to learn all the terms individually. We'll leave no stone unturned. We are not going to leave any stone which is unturned. We'll learn everything. Let's see, what is syngamy? Types. So the next one comes out to be the types, that is syngamy. Shall we go with this, students? Okay. Complete and permanent fusion of the male and female gamete. That is the complete or the permanent fusion of the male and the female gamete to form the zygote can be easily done. You see how nicely with the microscopic view they are able to see the egg cell. Okay. And this how is small is the sperm, and where after fusion, once the fusion has already happened, then the zygote formation is obvious. Okay, this is how the zygote is formed, where the gametic fusion can be seen. In case of the zygote, male is formed in the female egg, or the ova is going to match up. It's going to mate over there. Sexual reproduction by endogamy. I said you none of the stone will be left unturned. Endogamy. Types of endogamy, we have got syngamy, then we have got the endogamy. Example comes out to be is the Taenia solium. Taenia solium is nothing but the flatworm. Taenia solium comes out to be the flatworm. As you see in the diagram also, it's an absolute flatworm, what you can see over there. So it's going to, like a flatworm, you can see how big they can be. And the last end is called as the tail part. That is from the tail part we find that. From the tail part, students, we are going to find that through the tail part, the complete end part of the tapeworm will be released, and it shows a reproduction over there. Welcome to all the newcomers who have come to the class. We are discussing reproduction in organism, class 12th first portion. We are doing, students. We are discussing with that class 12th first portion. We are discussing today. We again have a class, all newcomers. We again have a class from 8 p.m. to 9 p.m. From the 8 p.m. to 9 p.m. again we have a class, and you can all join in that time. Next, what do we see? Sexual reproduction type of exogamy. Exogamy means gamete coming outside the gametogonial structure. Gamete coming outside the gametogonial structure is called as exogamy. So these are the gamete coming outside during the intercourse or gametogonial period. So gamete chooses the partner I hope this part is done to all of you have understood. It is seen outside the group. Example comes out to be is the rabbit. You see the best example is white rabbit. How beautiful these creatures looks, right, students? They look so awesome, right? Similarly, sexual reproduction types of hologamy we'll see. Hologamy was also one part we have seen. Hologamy, the fusion of the two organisms which is acting as a gamete, can be seen in case of your yeast. One of the best example comes out of yeast. You see, in case of the yeast, we can easily find out that in the yeast, budding yeast, that's the reason we call it as a budding yeast. It starts budding and there is a complete two demarcation of the sexual life cycle, you can be seen. Then the daughter cells or the non-parental cell, if they want to have the fusion with the mother, well, that is not so much easy. Okay, exchange of the fusion of the complete, that is the gamete, is going to take place in such a situation. Okay, next. Sexual reproduction. Now this is the image of a live spirogyra. I'll just zoom it so that it will be more clear to all of you. So how do we see over here? Students, we have a next class who have joined right now. We again have a class that is from 8 p.m. 9 p.m. Shall we go next, students? You see, in case of the spirogyra, as you see, How do beautifully they have seen. You see, one thing which is important is it is septate structure. This is one of the septate structure what we are going to see, student. And then also we find that through this septate structure, it is also going to possess the complete, that is the chloroplast. And in case of Spirogyra, why do we call it as Spirogyra? Because their chloroplast is spiral. So we can write over here, the chloroplast is spiral. Chloroplast is spiral. Spiral chloroplast it contains. Next we can see over here, that is the scalariform conjugation. A best example you can find lateral conjugation. We have already seen how the lateral conjugation takes place. Scalariform conjugation, you see how beautifully it shows where the genetic material is going to pass into the body through the scalariform structure with the use of a scalariform structure. You can see right over here with the scalariform structure, this is going in. Right over here, the scalariform structure, it is going in, students. Have you all understood till here? And you see conjugation, it's a conjugation tube which is going to be formed. It's a complete conjugation tube which is going to be formed, students. And through the conjugation tube, it is inserting inside. Through the conjugation tube, we find that it is going to be forming this structure. I hope it has been understood to all of you. Beta Anjali, you have joined a little late today. No problem, we'll have a class from 8 o'clock. Okay, again we'll have a class from 8 o'clock. That day, that time you can join, 8 p.m. Today, 8 p.m. today. You can join and we'll have. So this is a conjugation tube which is going to be formed, student. I hope you people are all liking the diagram because the diagrams are really important. And with a lot of effort, I'm trying to get the diagrams for all of you. And that will be, which is going to be useful for every one of you. So diagrams are really important, students. Okay. Thank you all of you. We'll meet in the next class. Till that time, thank you very much. Yes. You people need not to pay a single rupee from your pocket, and you can utilize completely the classes, and it will be very useful for every one of you students if you follow the classes. Similarly, every day I am taking classes for the students on UNACADEMY. Generally, I follow the timing, that is from 3 to 4 and 8 to 9. 8 to 9 is a fixed time, students, I'll tell you. Night 8 to 9, 8 p.m. to 9 p.m. is a fixed timing I take classes for UNACADEMY students, and that will be very beneficial. So what I have started doing this, I'll be recording the sessions and putting on YouTube, but if you come for a live class, it will be really interactive and useful for all of you. So if you come for the classes, this will generally help every one of you, and this will be properly guiding you through. It will be a guide post, it will be guiding all of you and will be helping you attain good marks in the coming exam. So let's start it, let's crack it in this week. Prepare nicely for the 2021 or class 12 student as well as class 11 student. We are going to prepare in this way, and I make sure that all students are going to get good marks. So all the best and all of you, and with UNACADEMY, let's crack it. Thank you all of you.